You may have learned to classify a rose or a human as living, rock and water as non-living. But why is this so? If you say that living is something that moves, but then flowing water can be called living, right? That's not entirely true. Life has a set of properties that an organism has to follow to be called living. And a wonderful discipline of science studies life in great detail. It is called biology. The word bio means living, and logi means study. So biology means the study of living things. Simply put, anything which interacts with its environment, use energy and reproduce are known as living things. For example, we humans eat food to get energy, feel and respond to our environment, and reproduce to make more of ourselves. Many other living things like insects, birds, and animals have the same properties like us, so they are all living organisms. Now the question arises, how many types of living things are there? And how are they different from us? Biology is a field which answers these questions and many more questions related to living things. What is biology? There are 7 characteristics that a living thing follows, and therefore we study this in the vast field of biology. These are 1. Order. If you examine a rose from near, you will notice it is very ordered. The petals are whorled around each other with the center bearing seeds. This illustrates the highly ordered structure that typifies life. Living cells are the basis of this complex organization. 2. Reproduction. Organisms reproduce their own kind. A mother giant panda will always give birth to a panda, not a horse. We learned that from Kung Fu Panda too. 3. Growth and development. Inherited information in the form of DNA controls the pattern of growth and development of all organisms. You most probably look like your parents or a close relative, thanks to the genes. 4. Energy processing. We humans eat food to get energy. The food we eat has also eaten some food to utilize its chemical energy stored in order to power its own activities and chemical reactions. 5. Response to the environment. All organisms respond to environmental stimuli. This is why you laugh when you are tickled. 6. Regulation. Many types of mechanisms regulate an organism's internal environment, keeping it within limits that sustain life. This is also called homeostasis. A lemur sunbathes to help raise the animal's body temperature on cold mornings. 7. Evolutionary adaptation. Lions use camouflage to blend in with the savanna, so they can easily hunt down an animal. Such adaptations evolve over many generations. Some new concepts like genes and DNA are introduced in this video. These will be explained in our upcoming videos. Stay tuned. What is diffusion? Ever wonder how substances move around the body, or how a metabolite gets in and out of the cell? Or how perfumed spray spread the aroma around in a matter of seconds from the corner you spray it in to the rest of your room? Diffusion is to be blamed. Yes, it is the kind of transport in which dissolved substances or particles move from region of higher concentration to lower concentration with no external energy involved. Since this transport does not require energy from external source, it is also called passive transport. Diffusion occurs when particles, gases, or dissolved substances are mobile or free to move as they spread from where they are in high concentration to the region where they are in low concentration throughout the concentration gradient. The concentration gradient is an environment in which particles are distributed in different concentrations, keeping high concentration region on one side and low concentration on the other side. In the human body, the blood takes oxygen using this simple mode of passive transport, in which oxygen moves from alveolar air spaces in which it happens to be in higher concentration, to the blood where it is used to be in lower concentration. It can also be conceived as the mode of transport in which substances move from region of abundance to the region of demand, where their concentration is lower. Cells use a bit different, yet sophisticated kind of diffusion. They use some carrier proteins to take in the required substance of nutrients, and since this mode involves facilitation of these proteins, to allow these substances inside the cell through the membrane. This kind of diffusion is called facilitated diffusion. An important example is that of blood. Oxygen binds with red blood cells in the bloodstream through facilitated diffusion, which gives us oxygenated blood. Cell. Have you ever examined a leaf under the magnifying glass? You may have noticed extremely small rectangular compartments in them. Did you know these keep the plant alive? And these small compartments are not just present in plants, but animals and microbes too. They are responsible for life and are called cells. Cells are the basic unit of life. Our today's guest is a cell. Please join your hands together as we invite him for a talk. Hello, I am a cell, and I am the smallest unit of every living organism. I can perform structural and functional roles in living organisms. I am different in eukaryotic and prokaryotic organisms. In eukaryotic cells, I have membrane-bounded organelles, like nucleus, that define cells' genetic makeup. Mitochondria provides energy. Endoplasmic reticulum help in protein folding. Golgi apparatus do protein packaging. Ribosomes help in protein synthesis. Lysosomes destroy the foreign objects, and the vacuole acts as a storage system of a cell. All these are termed organelles. All these organelles float in the cytoplasm, and substances like salts may be dissolved in it. This gives it a jelly-like appearance. The cytoplasm is also continuously streaming like waves in a calm lake. In short, I am like a fully functional factory, with different departments following different functions to make it run smoothly. In prokaryotes, I don't have membrane-bounded organelles. My nucleus is freely suspended in cytoplasm. In case of plants, I am surrounded by a blanket called cell wall, giving me strength, and then by cell membrane. I have a large vacuole located in center and a small nucleus at a side. In animal cells, a large nucleus is present in center, and small vacuole is present at a side. I don't have a cell wall. I only have cell membrane. You yourself are made up of millions of different types of animal cells. Can you name some of them? Branches of biology. Hey there, I am biology, and I have knowledge about all living organisms. And I am mother of 12 children. My first child is cellular biology, that studies the structure and functions of cells. The other one, molecular biology, studies the biological processes on a molecular level. Evolutionary biology solely focuses on biology from an evolutionary point of view, to predict how biology will evolve. Bioengineering builds products by using engineering concepts applied to biological systems. Cardiac stunts are an example of this. Marine biology is responsible for studying all the creatures that live within the marine environment. Medical biology studies all fields that are related to illnesses, alterations, and functions of the human body. Mycology studies solely fungi. Zoology is responsible for the study of animal life as a whole. Botany, it deals directly with the study of plants. Microbiology deals with the study of organisms that are invisible to naked eye. Here is environmental biology, which deals with the way in which all the living and non-living elements of the environment interact with each other. Last, but never the least, this is astrobiology, the branch of biology concerned with the effects of outer space on living organisms and the search for extraterrestrial life. What is your favorite branch of biology? Comment below and we will tell you more about it.
Necrosis versus apoptosis. Hello everyone. This tutorial will explain to you the main difference between apoptosis and necrosis. Introduction. All cells in a multicellular organism undergo growth and death. Cellular death is essential for an organism to grow and survive. Human body is made up of innumerable types of cells in various parts of the body. There are mainly two types in which a cell undergoes death, either through exposure to harmful environment, injury, or through a pre-planned and regulated process of disintegration. Cell death occurs via two different processes, one being necrosis and the other being apoptosis. Now the question is, what is necrosis? Necrosis is a process of cellular death, which occurs when cells are exposed to extreme conditions, which are very different from normal living conditions. This leads to damage to the internal cellular environment and rapid cell and tissue damage. So the basic inability of the cell to maintain balance when exposed to rough conditions leads to complete discord in the cell's functioning and eventually its death. What is apoptosis? Apoptosis is a pre-planned regular process of cell death occurring in the body where the cell itself actively takes part in its own death. It occurs as part of normal cellular multiplication and turnover. so cells die naturally to maintain a balance of the cellular multiplication. Apoptosis is required for smooth functioning of the body. Most cells in the body have a shelf life. For example, red blood cells live for 120 days. At the end, they are destroyed in the body. This is a method of apoptosis. Difference in cause and presentation. Necrosis is a pathological process which is detrimental to the body. It occurs when the cells are exposed to toxins or on exposure to extreme conditions, which could be anything from increased temperature, decreased oxygen levels, etc. These conditions lead to damage to the wall or membrane of the cell. An inability to maintain normal functioning in these conditions leads to an imbalance in the internal environment of the cell. This leads to inflammation and tissue damage, which leads to accumulation of cellular debris. Apoptosis is a normal, natural process which is necessary for the body as it maintains a balance in the number of cells in the body. If the cells do not die on time, it can lead to a tumor or cancer formation, which is an accumulation of unwanted cells. If cells die too fast, it can lead to conditions such as AIDS, heart disease, and liver disease. So if apoptosis does not take place as it should, there will be signs of disease. Difference in structural changes. In necrosis, there is a loss of integrity of the wall of the cell, which leads to swelling of the cell's contents as well as disintegration of small bodies of the cell. In apoptosis, there is no disintegration of the cell membrane or wall, rather there is a shrinkage of cellular contents along with clumping of the membrane. This leads to formation of condensed bodies known as apoptotic bodies. These bodies are recognized by the immune system of the body and destroyed efficiently by the immune cells. Thus, it is a well-coordinated process due to which there is no accompanying inflammatory reaction. Difference in biochemical reactions. Necrosis is a passive process which does not require energy and can occur at any point of time. This is a random event which is unregulated. Apoptosis is an active process which requires energy and occurs in an organized manner. Many enzymes and agents are required to carry out various steps of this process. Summary. Apoptosis and necrosis are variants of cell death that vary greatly in all ways except the end result. Necrosis is a type of cell death wherein the cell dies an untimely death due to uncontrolled external factors, and apoptosis is a predefined cell suicide wherein the cell actively destroys itself to maintain smooth functioning of the body. Apoptosis is a natural physiological process which is required by the body, while necrosis is a pathological process which stems from inflammation and tissue damage.
Mitosis, when the cell splits apart. What is mitosis? Eventually, cells need to duplicate. There are two main methods of replication, mitosis and meiosis. This tutorial will talk about mitosis. The big idea to remember is that mitosis is the simple duplication of a cell and all its parts. It duplicates its DNA, and the two new cells, daughter cells, have the same pieces and genetic code. Two identical copies come from one original. Beyond the idea that two identical cells are created, there are certain steps in the process. Before a cell divides, it is in resting phase. The cell in resting phase is not actually resting, since it has to maintain its reservoir before it undergoes division. This phase is called interphase. It prepares the cell for division, i.e. protein synthesis, DNA duplication, and enzymes. After this phase comes mitosis, which is further divided into karyokinesis, division of nucleus, and cytokinesis, division of cytoplasm. Karyokinesis involves the four basic steps of mitosis that are prophase, metaphase, anaphase, telophase. Here we start with prophase. The chromosomes start coiling and become condensed. Nuclear envelope disintegrates. Gene transcription ceases during prophase and does not resume until late anaphase to early G1 phase. The nucleolus also disappears during early prophase. Centrosome gives rise to mitotic apparatus, i.e. generation of spindle tubules. Metaphase. Now all the pieces are aligning themselves for the big split. The DNA lines up along a central axis, and the DNA, chromatin, has now condensed into chromosomes. Two strands of chromosome are connected at the center of the centromere. The tubules connect to the centromere, not the DNA. Anaphase. Here we go. The separation begins. Half of the chromosomes are pulled to one side of the cell. Half go the other way. When the chromosomes get to the side of the cell, it's time to move on to telophase. Telophase. Now the division is finishing up. This is the time when the nuclear envelope begins to synthesize again along the separated chromosomes. Cytokinesis pinches the cell membrane in the middle. Cell membrane closes in and splits the cell into two. Now we have two separate cells, each with half of the original DNA. Up till now, we have discussed what is mitosis and its different phases. Now it's important to know about significance of mitosis. Significance. It is an equational division through which identical daughter cells are produced, having the same amount and type of genetic constitution as that of the parent cell. It is responsible for growth and development of multicellular organisms from a single-celled zygote. The number of chromosomes remains the same in all cells produced by this division. Thus, the daughter cells retain the same character as those of the parent cell. It helps the cell in maintaining proper size. Mitosis helps in restoring wear and tear in body tissues, replacement of damaged or lost part, healing of wounds, and regeneration of detached parts, as in the tail of lizards. It is a method of multiplication in unicellular organisms. If mitosis remains unchecked, it may result in uncontrolled growth of cells, leading to cancer or tumour. The next important phase of cell cycle is meiosis.
Blood groups and blood transfusion disorders of blood. What is blood? There are various definitions which defines blood. In simple words, it's a red liquid which circulates in arteries and veins, humans and other vertebrate animals, carrying oxygen to and carbon dioxide from the tissues of the body. Other defines blood as vital principle of life. It is the mechanism of blood in bodies of living organisms which can differentiate one individual from the other based on the presence or absence of specific antigens in blood. This system is called blood group. There are 4 blood groups: A, B, AB, and O blood groups. While blood types are 100% genetically inherited, the environment can potentially determine which blood types in a population will be passed on more frequently to the next generation. Blood type A, if the red blood cell has only A molecules on it. Blood type B, if the red blood cell has only B molecules on it. Blood type AB, if the red blood cell has a mixture of both A and B molecules. Blood type O, if the red blood cell has neither A or B molecule. Blood group facts. There are 8 different common blood types, which are determined by the presence or absence of certain antigens, which are substances that can trigger an immune response if they are foreign to the human body. Since some antigens can trigger a patient's immune system to attack the transfused blood, safe blood transfusions depend on careful blood typing and cross-matching. Now, the next turn is to know about an interesting factor known as Rh factor, which can be either present positive or absent negative. In general, Rh negative blood is given to Rh negative patients, and Rh positive blood or Rh negative blood may be given to Rh positive patients. Are you a universal donor? A universal recipient? The universal red cell donor has type O negative blood type. The universal plasma donor has type AB positive blood type. Blood transfusion. Blood transfusion is a medical term. It means a procedure used to transfer blood, or some products based on blood, from the circulatory system of one human to that of another human. Blood types are very important when a blood transfusion is necessary. In a blood transfusion, a patient must receive a blood type compatible with his or her own blood type. If the blood types are not compatible, red blood cells will clump together, making clots that can block blood vessels, causing a potentially fatal situation. Therefore, it is important that blood types can be matched before blood transfusions take place. In an emergency, type O blood can be given because it is most likely to be accepted by all blood types. However, there is still a risk involved. Why blood transfusions are performed? Loss of blood during surgery or from an injury or an illness. An inability to make enough blood. Some illnesses and treatments can harm the bone marrow's ability to make blood, e.g., chemotherapy decreases production of new blood cells. To prevent complications from an existing blood or bleeding disorder, such as sickle cell disease, thalassemia, or anemia caused by kidney disease, haemophilia, or von Willebrand disease. Preparing for a blood donation. If your child needs a blood transfusion, the doctor will speak to you about the procedure. If you have questions, be sure to ask. When you feel comfortable with the information and your questions have been fully answered, you'll be asked to sign an informed consent form. This form states that you understand the procedure and its risks, and you give permission for your child to have the blood transfusion. If the situation is not a life-threatening emergency, 2 tests will be done before the transfusion: blood typing and cross-matching. Have you ever wondered why you are different from your siblings? The answer lies within the amazing process of meiosis. What is meiosis? Meiosis is a process where a single cell divides twice to produce 4 cells containing half the original amount of genetic information. These cells are our sex cells: sperm in males, eggs in females. During meiosis, one cell divides twice to form 4 daughter cells. These 4 daughter cells only have half the number of chromosomes than the parent cell. They are haploid. Meiosis takes place in germ or sex cells called gametes, eggs in females and sperm in males. Similar to mitosis, cells also pass through the interphase, G1, S, and G2 phase before they enter meiosis. Here are the distinct phases of meiosis. Interphase, meiosis 1, cytokinesis 1, meiosis 2, cytokinesis 2. First at interphase. In interphase, DNA is copied, resulting in 2 identical full sets of chromosomes to prepare for division. Outside of the nucleus are 2 centrosomes, each containing a pair of centrioles. These structures are critical for the process of cell division. In next step at meiosis 1, meiosis 1 takes place in following steps. Prophase 1. Prophase 1 is typically the longest phase of meiosis. During prophase 1, homologous chromosomes pair and exchange DNA, homologous recombination. The new combinations of DNA created during crossover are a significant source of genetic variation and result in new combinations of alleles, which may be beneficial. The paired and replicated chromosomes are called bivalents or tetrads, which have 2 chromosomes and 4 chromatids, with 1 chromosome coming from each parent. The process of pairing the homologous chromosomes is called synapsis. At this stage, non-sister chromatids may cross over at points, called chiasmata, plural, singular chiasma. Prophase 1 has historically been divided into a series of substages, which are named according to the appearance of chromosomes. Leptotene. In this stage of prophase 1, individual chromosomes, each consisting of 2 sister chromatids, become individualized to form visible strands within the nucleus. Leptotene is of very short duration, and it's when the progressive condensation and coiling of chromosome fibers takes place. Zygotene. Chromosomes approximately line up with each other into homologous chromosome pairs through synaptonymal complex. The paired chromosomes are called bivalent or tetrad chromosomes. Pachytene. This is the stage when homologous recombination, including chromosomal crossover, crossing over, occurs. Non-sister chromatids of homologous chromosomes may exchange segments over regions of homology. At the sites where exchange happens, chiasmata form. Diplotene. The synaptonymal complex degrades and homologous chromosomes separate from one another a little. However, the homologous chromosomes of each bivalent remain tightly bound at chiasmata. The regions where crossing over occurred, the chiasmata remain on the chromosomes until they are at the transition to anaphase 1. Diakinesis. It closely resembles prometaphase of mitosis. The nucleoli disappear, the nuclear membrane disintegrates into vesicles, and the meiotic spindle begins to form. Synchronous process. During these stages, two centrosomes, containing a pair of centrioles in animal cells, migrate to the two poles of the cell. The microtubules invade the nuclear region after the nuclear envelope disintegrates, attaching to the chromosomes at the kinetochore. The kinetochore functions as a motor, pulling the chromosomes along the attached microtubule. Microtubules that attach to the kinetochores are known as kinetochore microtubules. Other microtubules will interact with microtubules from the opposite centrosome. These are called non-kinetochore microtubules or polar microtubules. Metaphase 1. Homologous pairs move together along the metaphase plate. The paired homologous chromosomes align along an equatorial plane that bisects the spindle. Anaphase 1. Kinetochore microtubules shorten, pulling homologous chromosomes, which consist of a pair of sister chromatids, to opposite poles. Non-kinetochore microtubules lengthen, pushing the centrosome further apart. The cell elongates in preparation for division down the center. Telophase 1. The first meiotic division effectively ends when the chromosomes arrive at the poles. Each daughter cell now has half the number of chromosomes, but each chromosome consists of a pair of chromatids. The microtubules that make up the spindle network disappear, and a new nuclear membrane surrounds each haploid set. The chromosomes uncoil back into chromatin. Cytokinesis, the pinching of the cell membrane in animal cells or the formation of the cell wall in plant cells occurs, completing the creation of two daughter cells. Sister chromatids remain attached during telophase 1. Cells may enter a period of rest known as interkinesis or interphase 2. No DNA replication occurs during this stage. Meiosis II. Meiosis II is the second meiotic division and usually involves equational segregation or separation of sister chromatids. Mechanically, the process is similar to mitosis, though its genetic results are fundamentally different. The end result of meiosis II is production of 4 haploid cells, N chromosomes, 23 in humans. The 4 main steps of meiosis II are prophase II, metaphase II, anaphase II, and telophase II. In prophase II, we see the disappearance of the nucleoli and the nuclear envelope again, as well as the shortening and thickening of the chromatids. Centrosomes move to the polar regions and arrange spindle fibers for the second meiotic division. In metaphase II, the centromeres contain 2 kinetochores that attach to spindle fibers from the centrosomes at opposite poles. This is followed by anaphase II, in which the remaining centromeric cohesion is cleaved, allowing the sister chromatids to segregate. The sister chromatids by convention are now called sister chromosomes as they move toward opposing poles. The process ends with telophase II, which is similar to telophase I and is marked by the disassembly of the spindle and decondensation and lengthening of the chromosomes. Nuclear envelopes reform and cleavage or cell plate formation eventually produces a total of 4 daughter cells, each with a haploid set of chromosomes. Meiosis is now complete and ends up with 4 new daughter cells. Enzymes. Human body is a product of different chemical reactions and processes, but what controls these reactions? In 1833, a French chemist, Anselme Payen, was the first to discover the vital force that drove these reactions and named it enzyme. Enzymes are substances, proteins, or in some cases, ribonucleic acid, RNA, that speed up a biochemical reaction by modifying specific substances called substrates. Enzymes are supremely selective in who they bind to and modify, hence their specificity. Mode of action. Easier said, but how does a tiny enzyme speed up a chemical reaction? The answer to this is simple. Enzymes perform this critical task by lowering a reaction's activation energy, that is, the amount of energy needed for the reaction to begin. Enzymes bind to their substrates, hold them in such that chemical bond breaking and bond forming processes take place more easily. Enzymes have a specific place in them called active site, where the substrate binds and real-time action takes place. Active site has a specific size, shape, and chemical behavior rendered to it by specific arrangement of amino acids. Thanks to these amino acids, an enzyme's active site is unique only to a particular substrate. In addition to the active site, many enzymes consist of a non-protein part called cofactor. Cofactors may be cations, positively charged metal ions, activators, bound temporarily to the active site to activate the enzyme. Organic molecules, vitamins or vitamin products, coenzymes that join enzyme-substrate complex temporarily. Prosthetic groups, permanently enzyme-bound. Many enzymes only perform their catalytic role when associated a coenzyme. An inactivated enzyme, protein, along with its coenzyme, non-protein, makes up a system called holoenzyme. To add to the complexity, scientists termed this inactivated enzyme as apoenzyme. And therefore, the equation becomes holoenzyme equals apoenzyme plus coenzyme. Models of enzyme action. Lock and key hypothesis. This is the simplest model to represent how an enzyme works. The substrate simply fits into the active site to form a reaction intermediate. Just like the key fits in its specific lock, the shape isn't changed here. Rather, the structure of the substrate absolutely complements the structure of the enzyme, like puzzle pieces. Induced fit hypothesis. In this model, the enzyme, upon binding of substrate, changes shape. The matching between an enzyme's active site and the substrate isn't just like two puzzle pieces fitting together. Rather, the enzyme changes shape and binds to its substrate even more tightly. This fine-tuning of the enzyme to fit the substrate is called induced fit. Environmental effects on enzyme function. Active sites are very sensitive. They sense even the slightest change in the environment and respond accordingly. Some of the factors that affect the active site and consequently enzyme function include temperature. The suitable temperature for enzymes to function properly is 37 degrees Celsius. Increasing or decreasing the temperature above 37 degrees Celsius affects chemical bonds in the active site, making them less suited to bind substrates. Higher temperatures denature enzymes. pH. Amino acids present in the active site are acidic or basic. Fluctuation in pH can affect these amino acids, making it hard for substrates to bind. Extreme pH values can denature enzymes. Enzyme concentration. Increasing enzyme concentration will increase the rate of reaction, as more enzymes will be available to bind with the substrates. However, after a certain concentration, any increase will have no effect on the rate of reaction. Substrate concentration. Increasing substrate concentration increases the rate of reaction. This is because more substrate molecules will be colliding with enzyme molecules, so more product will be formed. But again, this effect is valid up to a certain concentration. Inhibition of enzyme activity. Some evil substances called inhibitors reduce or even stop the activity of enzymes in biochemical reactions. They either block or distort the active site, thus inhibiting the reaction. Based on this, there are two types of inhibitors given below. Competitive inhibitors occupy the active site and prevent a substrate molecule from binding to the enzyme. Non-competitive inhibitors attach to parts of the enzyme other than the active site to distort the shape of an enzyme. General structure of leaf. Have you ever wondered when you look at a plant, what is the most abundant thing you observe? Yes, it is the leaf. A leaf is the main organ of plant, which is used to carry out the vital process of photosynthesis. We have learned in our previous videos that photosynthesis is the process in which sunlight is used to convert the very basics of environment, i.e. carbon dioxide and water, into carbohydrate sugars, along with rich, fresh air by the oxygen produced. But how is it leaf become the most appropriate organ for this process? Adaptation of leaves for photosynthesis. Leaves grow to be highly functional for processing sunlight, mineral and gases thanks to these features. Large surface area to capture more light and chlorophyll to absorb it. Veins to support the structure and allowing the flow of xylem and phloem. Thin structure and stomata to allow the diffusion of carbon dioxide. Parts of leaf. Lamina, or the leaf blade, i.e. the widest part of leaf. Petiole, attaching leaf with the main stem. Midrib, divides to give rise to veins. Veins, as bundles of vascular tissues. Margin, the edge of leaf. Internal structure of leaf. The internal structure of leaf consists of epidermis, stomata, cuticle, mesophyll, chloroplast, vascular tissue. Now let's have a deeper insight of these parts. Epidermis. The outermost cell layer of the leaf is known as the epidermis. It is one cell thick in most plants. It is divided into two, upper and lower epidermis. The function of epidermis is to provide protection to internal tissues and allow the exchange of gases via stomata. Stomata. Stomata are the openings which allow for this exchange of gases. There are two guard cells located on either side of stomata to regulate its opening and closing. The guard cells are the only cells in the epidermis layer which contain the chloroplast. Cuticle. Cuticle is a waxy layer which covers the surface of a leaf. The waxy layer reduces the water loss from leaf surface. Some leaves may have trichomes, which are the small hairs on leaf surface. They have toxic compounds or bad-tasting chemicals, which reduces the chance of them being eaten. Also, they reduce rate of transpiration by blocking airflow across the leaf surface. Mesophyll. In some plants, layers of cells known as mesophyll are present. This layer have two different arrangement of cells, palisade parenchyma and spongy mesophyll. Palisade parenchyma cells are column-shaped cells, which are tightly packed. They help in carrying out photosynthesis. In contrast, spongy mesophyll are loosely arranged cells below the palisade parenchyma. The spongy mesophyll have air spaces among them, as they are loosely arranged, which allows gaseous exchange between outer environment and leaf. In both layers of mesophyll, many chloroplasts are present. Chloroplast. Chloroplast contains chlorophyll pigment that captures energy required for photosynthesis to happen. This ability to capture energy and produce food earned chloroplast the title as the powerhouse of plant. Chloroplast can be found moving around within the cells of mesophyll. There are usually 30 to 40 chloroplasts per mesophyll. Vascular tissue. Vascular tissues make up the plant's circulatory system, much like our own. In humans, blood transports oxygen and water to the body. In plants, xylem and phloem plays this role. Xylem transports water and minerals to leaves and consists of tracheids and vessels. Phloem is responsible for transport of photosynthetic products to other parts of plant.
Stem cells. When hearing the two words together, stem cells, most of us would think of cells residing inside the stem of plants. But in reality, stem cells are cells that divide to produce specialized other cells in order to replace the worn-out ones and maintain a homeostasis. Think of it like a branch of a tree on which leaves, fruits and small branches emerge. Stem cells work just as similar. Specialized cells are differentiated from its precursor stem cells to maintain the body's internal environment. Until now, two kinds of stem cells of human sources have been known: embryonic stem cells and adult stem cells. Embryonic stem cell has the ability to differentiate into any necessary type of body cells, while adult stem cell has limited differentiation of cells specific only to its tissue origin. To avoid the ethical issue in research, embryonic stem cell can be recreated by replacing the egg cell's haploid genome with adult's diploid genome, as in the case of the cloned sheep Dolly, or by introducing several iPS genes into an adult skin cell. The most common example a biology student could give of adult stem cell is that of blood cells. If we are hiking on the top of a snowy mountain, the rising demand for oxygen from our body is met as soon as our kidney responds by secreting a type of hormone into the blood. This particular hormone sends a signal to bone marrow for the differentiation of stem cells into more red blood cells to fulfill our body's need. Stem cells are also being cultured to create skin that can be grafted on burned bruises. Another example of cultured stem cell is cultured meat. The meat grown in a lab may be considerably more beneficial to our health and environment than conventional meat. In addition, plants are also possible to be created in labs by providing a specific amount of nutrients and growth factors for the undifferentiated, non-specialized stem cells to start generating shoots and roots. What is DNA? Every living thing has DNA, from plants to animals to bacteria, even to viruses which were once considered non-living. And most importantly, we humans have it. DNA stands for deoxyribonucleic acid, and although every living thing has DNA, its varied compositions differentiate you from others. The interesting fact is that humans' DNA is 99% the same. Therefore, it is only 1% of DNA that makes you unique. Does this mean twins don't have exactly the same DNA? No, they don't, unless they're monozygotic or identical twins. So now the questions arise. Why every living thing has DNA? What is so important in it? And what does it do? DNA has a recipe for synthesizing proteins. DNA synthesizes RNA, or ribonucleic acid, by a process called transcription. It is further translated into proteins by a process termed as translation. The explanation of DNA conversion into RNA, and finally RNA into proteins, is called central dogma. DNA contains hereditary information that is passed on to you from your parents. If you and your father have the same colour of eyes, or your hair are as silky as your mother, it is because of DNA that you inherited from your parents. Even many diseases are passed on from generation to generation because of DNA. Where does DNA reside in your body? Your body is made up of billions and trillions of cells, and almost all cells have DNA in their nucleus. Can you tell us which cell may not contain DNA? Now let's talk about the structure of DNA. Think of DNA structure as a ladder whose rungs are made up of different bases and sides as sugar phosphate. Each composition of this nitrogenous base, pentose sugar, and phosphate group is called nucleotide. The nitrogenous bases are of 4 different types: adenine, guanine, cytosine, and thymine. Adenine will always bind with thymine, and cytosine with guanine. This type of bonding is hydrogen bonding. Phosphate and sugar are linked together to make backbone, and complementary bases are attached on it. Due to the bond angle of the sugar-phosphate molecules, the linkages will eventually form a double helix structure. There's a lot more to know about DNA, but that's all for now.
In 1779, a scientist observed that in the presence of light, plants give off bubbles from their green parts, while, in the shade, the bubbles eventually stop. He identified the gas as oxygen. He also observed that, in the dark, plants give off carbon dioxide. This man was Jan Ingenhousz, and this is how he became the first man to discover the process of photosynthesis. You may already know that photos is the Greek word meaning light, and synthesis is another Greek word meaning putting together. However, what is photosynthesis and why is it important?
This is the process of making glucose, which is the energy source for most cell. It makes this from sunlight energy, water, and carbon dioxide. Only plant cells can do this. The special organelle within the plant cells responsible for this process is called chloroplast. Photosynthesis goes through 2 stages. Stage 1. Light-dependent reaction. This reaction depends on the presence of light. It starts when photons from sunlight strike the leaf, enter all the way through to the chloroplast and its thylakoid discs, excite the chlorophyll and activate electrons. During this process, water is split into oxygen and hydrogen ions, releasing electrons. This is how the electron in the chlorophyll gets replenished and where the oxygen generated during photosynthesis comes from. The activated electrons then move through a series of electron carriers, also referred to as electron transport chain, and in this process leads to the accumulation of the hydrogen ions, generating a proton gradient inside the thylakoid compartment. As the protons decrease their concentration gradient through the ATP synthase, ADP and PI come together to form ATP. The adenosine triphosphate, or the ATP, is an energy molecule that cells exchange, like currency, to release energy. The electrons going through the electron transport chain eventually end up combining with NADP+ to form NADPH, another energy-rich molecule that will be used later. Thus, the result of the light reaction is the production of oxygen released from the leaves, ATP and NADPH. The main difference between ATP and NADPH is the amount of energy molecules they release. Stage 2. Light-independent or dark reaction. This reaction can proceed in the absence of light, but calling it a dark reaction might be misleading as it doesn't happen only in the dark. It can just as well happen in the light. This process uses the energy from light reaction to convert carbon dioxide into glucose. This might sound simple, but in fact, the conversion of carbon dioxide to glucose proceeds through a series of reactions that start with 3-ribulose bisphosphate, RUBP, and eventually end up with the same molecule, producing glucose in the process. Because these series of reactions start and end with the same molecule, they are referred to as a cycle. Specifically, the Calvin cycle. The overall complete photosynthesis reaction will be needing 6 carbon dioxide molecules and 6 water molecules powered by the sun energy to eventually produce glucose and 6 oxygen molecules. As long as a plant remains green, it can benefit our environment in many ways and also transfers the energy to us when we eat it. Comment below to let us know if you would like us to cover the Calvin cycle in an upcoming video. All living organisms, plants need nutrients for their proper growth and development. They get carbon, oxygen and hydrogen from air and water to carry out photosynthesis, which is a very important process for plants. But what about mineral nutrients? Where do they get their mineral nutrients from? The answer is soil. The presence of minerals in soil is a result of weathering of parent rocks. This means the parent rocks undergo various chemical and physical mechanical changes, leaving behind these mineral elements in the soil as a result. These minerals in soil are either in absorbed form or in dissolved form. So to absorb the minerals from the soil, plants use their roots and in some cases their leaves. The mineral nutrients absorbed have many functions and roles to play in the body of plants. They are constituents of various proteins, pigments, nucleic acids and enzymes. They also play roles in cell signaling and metabolism. Plants take up these mineral nutrients in the form of ions. This uptake of minerals can either be a passive process, termed as passive transport, which takes place along the concentration gradient, thus requiring no energy, or an active process, termed as active transport, which requires energy because it occurs against the concentration gradient. The passive process transport or absorption can be explained by diffusion theory, the ion exchange theory, and Donan's equilibrium. Diffusion theory. When the concentration of mineral ions is higher in the external soil solution than the root, then the ions in the soil solution will move from a region of higher concentration, in this case the soil solution, to a region of lower concentration, that is the roots. This movement of molecules or ions is called diffusion. The ions keep moving inside until an equilibrium is reached. One of the reasons that allows the ions to diffuse into the root cells is because of the apparent free space. However, unlike non-charged molecules, ions will have difficulty to pass through the hydrophilic membrane directly. To facilitate the movement, the proteins called channel proteins and carrier proteins are needed. Ion exchange theory. In this process, the ions, mainly cations, that are absorbed to the root surface due to the negative charge of the cell wall, are exchanged with the ions present in soil. The positively charged hydrogen ions absorbed into the surface of the roots can easily be exchanged with cations of sodium and potassium present in the soil. Similarly, an anion like hydroxyl ion from the root cell can be exchanged with anion present in the soil. Ion exchange can be explained by contact exchange theory. According to contact exchange theory, ions can be taken up the plant without being dissolved in soil. The ions absorbed onto the root surface and the ions attached to clay particles are not entirely static. They oscillate in a small volume of space, and if the roots of plant and the soil particles are in close proximity that the oscillation volume of the ions overlap, ion exchange can take place. The exchanged ions enter the root by diffusion. Carbonic exchange theory. Roots of the plants continuously respire, which results in production of carbon dioxide. The carbon dioxide produced during respiration in roots combines with water to form carbonic acid, which dissociates into hydrogen and bicarbonate ion. The hydrogen ions produced as a result are exchanged with cations in soil. Donan equilibrium. Another theory of passive absorption is Donan's equilibrium theory, which states that there are some ions inside the root cells that are indiffusible across the membrane due to the selective nature of the cell membrane. If the root cells have cations accumulated inside as indiffusible ions, then anions present in soil enter the membrane without expenditure of energy to maintain a balanced electrical potential. The movement of anions in this example will result in an equilibrium which may not be equal in terms of concentration. These mechanics enable plants to take up minerals from the soil to ensure their proper growth. Generally, the mineral ion concentration inside the cell can be higher than the surrounding soil. In this case, the ions move against the concentration gradient. That is from lower concentration to higher concentration. This movement is termed as active transport and requires energy. Two theories, carrier concept theory and electrochemical gradient theory, have been proposed to explain how ions are taken up by plants via active absorption. Carrier concept theory. According to the carrier concept, the movement of ions across a membrane is mediated by special carrier molecules present in the membrane. These carrier molecules are usually proteins, but they can also be cytochromes and lecithin, phospholipids. Generally, carrier molecule works by binding ions to the carrier molecule and forms a carrier ion complex, causing a change in the conformation of the carrier molecule. The carrier ion complex passes through the membrane. When it reaches the inner side of the membrane, the carrier molecule changes its conformation and releases the ion. After the release of the ion, the carrier moves back to the outer surface of the membrane to bind another ion. The binding of ion and its release both require energy, which is provided by ATP. Electrochemical gradient hypothesis. An electrochemical gradient exists across the plasma membrane of the cells. This is due to two things. 1. The splitting of water molecules into hydrogen and hydroxyl ions. 2. The conversion of ATP into ADP and PI, inorganic phosphate. The proton pumps, in plasma membrane, pump out hydrogen ions while hydroxyl ions stay inside the cell, giving inside of the cell a negative charge. The proton concentration increases outside the cell than inside, generating a proton motive force. The electrical and proton gradients established as a result drive cation and anion uptake. Anions from outside the cell are exchanged with hydroxyl ions inside the cell, while the cations move inside the cell via ATPase. ATPase configuration changes as it binds the cation. It then transports the cation into the cell and changes back its configuration to bind another cation. This process is aided by the hydrolysis of ATP. Importance of nitrates and magnesium ions. In 1939, Arnon and Stout introduced the term essential mineral element, along with the three main points that describe essentiality of an element. According to them, an element is essential if the plant is not be able to complete its life cycle in its absence. It cannot be replaced by another element, and it is directly involved in plants' metabolism. Based on plants' requirements, there are at least 17 elements known as essential nutrients. Two of the most important of them are nitrates and magnesium. Nitrogen and magnesium are both essential elements needed by plants to ensure their proper growth. Nitrogen in the atmosphere cannot be taken in by plants directly, so they absorb it in the form of nitrates and ammonium. In aerobic soils, plants absorb nitrates with the help of their roots, and once it enters, some of the nitrate is reduced to ammonium by enzymes. It is then incorporated into amino acids with the help of more enzymes. Nitrates, beside ammonium, serve as a source of nitrogen, which is a component of many proteins. Photosynthetic pigments, coenzymes, purines, pyridamines, vitamins, and protoplasm. Magnesium is another important element in plant nutrition. It is present in the center in the porphyrin structure of chlorophyll and also serves as an activator of enzymes involved in photosynthesis and respiration. It is also important in DNA and RNA synthesis.